So I'm going to be sharing my screen, just looking at the objectives for the lecture notes. So exam 3 will cover starting with fungi up through pathogenesis. So not non-specific defenses, not quorum sensing, but all the way up through pathogenesis. But we'll start off with the fungi, right? So compare and contrast fungi with true plants and bacteria. Of course, we know fungi are not plants, right? They don't have cellulose like plants do. Although some fungi may be green, that green is not chlorophyll, so they're not photosynthetic. No xylem, no phloem, right? So fungi are very, very different. Of course, they're eukaryotes, but they're not plants. And when it comes to bacteria, of course, we know bacteria are prokaryotes. So bacteria do not have a nucleus, no membrane-bound organelles. Of course, fungi have all of those because they're eukaryotic organisms. So I just want you to, think about some comparisons between things you know like plants and things you've learned like bacteria and fungi. And we do have 5 phyla that I talked about: Ascomycota, Zygomycota, Deuteromycota, Basidiomycota, Oomycota. And when it comes to the classification, of course, that's based on the sexual phase. What if they cannot find a sexual phase? Where do you put that fungus? Into what phylum? If there's no known sexual phase, then what phylum do you put that fungus into? Got a 20% chance.
Deuteromycota.
Right, the Deuteromycota, right? Which phylum has some carcinogenic toxins we have to worry about?
Is it the Ascomycota? Ascomycota.
Right, so the Ascomycota, an example Aspergillus we talked about, has aflatoxins. And the Basidiomycota are the thick fleshy things like mushrooms, which some of you like to eat. And the Oomycota has been reclassified into Protista, so no longer technically considered in the fungus kingdom. So molds and yeasts. Remember, yeasts are single-celled microorganisms. They do form colonies like bacteria. Lots of yeasts are slimy, and the molds are fuzzy and filamentous. Of course, they're both eukaryotes, but they just have different forms. Sometimes a fungus can be both a yeast and a mold. What do we call those kinds of fungi? That can exist as either a mold or yeast, depending on environmental conditions.
Vegetative?
Nope, that's just the normal living state. But what do we call the fungus that can be a yeast or a mold? It can switch back and forth. Two shapes.
Dimorphism?
Right, so dimorphism is a condition whereby an organism can exist in 2 different states. When it comes to spores, what can you tell me about the number of chromosomes in spores, fungal spores?
Are the spores I think they're haploid?
They're always haploid, right? And fungi produce a lot of spores because the primary function is reproduction, right? So fungi make a lot of spores. They're very lightweight, so they can easily be spread. They're nowhere near as tough as a bacterial endospore is. And remember, bacterial spores are for survival, not reproduction. But fungi use their spores for reproduction. And what's a mycosis? What's a mycosis?
Infection caused by a fungus.
Right, so some kind of fungal disease. And remember, it can be difficult to treat fungal infections because they're eukaryotes like us, right? When it comes to viruses, we started off, talking about some general characteristics of viruses and then focused on the bacterial viruses first, and then we got into animal viruses later. So remember, viruses are not technically living organisms. They are not cellular organisms. No cell walls, no cytoplasm, no organelle, no aerobic respiration, right? No translation, no protein synthesis, right? Because they're basically just inanimate, as opposed to bacteria, which are living cells, right? Cell walls, cell membranes, cytoplasm, so all that good stuff. Some viruses do have membranes. Where did those membranes come from?
A host cell
Right,
member.
the host cells. And those are typically animal viruses. But back to bacteriophages. So remember that the protein coat is called a capsid. It contains the nucleic acid, which for a T-even bacteriophage is DNA. We have complex viruses, which is what a T-even bacteriophage is. So remember, the complex virus looks like a spaceship, has a sheath and tail fibers, so we call that complex. In order to grow a bacteriophage, you need a host, and that host will be some kind of bacteria, right? Because viruses must be grown in living hosts. And all viruses follow the same general 5 steps, right, for replication: attachment, penetration, biosynthesis, maturation, and release. Specifically for the bacteriophages, like a T-even bacteriophage, it lands on the host cell, uses its tail fibers to attach to host cell receptors. Then the virus uses lysozyme to get through the bacterial cell wall. The sheath contracts and squirts the DNA inside. Once inside, that viral DNA takes over, right, the bacterial cell, forces the bacterial cell to replicate the viral DNA, to transcribe and translate it to make the viral proteins. Everything assembles. The viral DNA gets incorporated into the capsid. And then the viruses use lysozyme to completely rupture the bacterial cell wall so they can be released and float away to infect other hosts. So that was the lytic life cycle, right? The end result is the destruction or the lysis of the bacterial host cell. But some viruses are lysogenic. So remember, lysogeny is similar to dormancy, where the bacterial virus infects the bacterial cell, but it doesn't replicate itself, it just goes dormant. The example we used was lambdaphage, which when it injects its DNA, the DNA circularizes so it can recombine with the bacterial chromosome and integrate, forming a prophage. And then viral proteins keep the prophage dormant, so it's not transcribed and translated to make new viruses. And so that bacterial cell is fine, at least for a while, right? It's protected from other lytic phages. So remember, a bacterial cell that has a lysogenic phage in it is actually protected from lytic phages, right? Because it's already got a dormant phage inside. And then potentially the phage can reproduce itself a lot more because every time that bacterial cell divides, every daughter cell has a copy of the prophage. But we know at some point the prophage can leave. It can cut itself out of the bacterial chromosome. If that happens, the virus will follow a lytic life cycle, make more copies of itself, and kill that host cell. Now, some of the consequences of lysogeny, one of them, as I mentioned, was that the bacterial cell with the prophage is protected from lytic phages. But the prophage may carry a toxin gene with it. And we talked about the diphtheria toxin, botulism toxin, and the cholera toxin, which are encoded by bacteriophage genes. And that toxin gene actually makes those bacteria pathogenic. And remember, toxins are the things that help a pathogen to survive and spread within a host, and the toxins also do damage to the host. So that enables the bacterial cell to reproduce itself a lot, which means the dormant virus, that prophage, gets reproduced a lot because all their daughter cells have a prophage. Viroids are less than viruses. Viroids are simply naked pieces of RNA, a little short piece, 400 to 500 base pairs long. And these viroids are infectious, right? They're transmissible. They cause diseases so far only in plants. But it's just a little piece of RNA, and that's it. And what's less than RNA? Prions, right? So prions are proteins that are found in the brain. So there's normal prion proteins found in animals, especially mammals. But it's the abnormally shaped prion that causes the problem. The abnormally shaped prions cause neurons to die around them, and that leads to diseases called spongiform encephalopathies, like mad cow disease. And the prions are extraordinarily stable proteins, right? Have to burn them to ashes to destroy them. And so the kinds of things that can be infectious, remember we talked about the most complex things, which are eukaryotic cells, organisms like Plasmodium and Trypanosoma. And then the next are prokaryotes, right? They're simpler, right, than eukaryotes. And we talked about a lot of bacterial pathogens like Salmonella, Shigella, Mycobacterium tuberculosis, Mycoplasma pneumoniae. So we talked about a lot of different, right, bacterial pathogens. And what's less than a cell? A virus, right? Just a piece of nucleic acid surrounded by protein with or without a lipid membrane. And we talked about a lot of different animal viruses, all the herpes viruses, COVID, HIV. So we talked about a lot of different viruses. And then what's less than a virus is a viroid, right? That little naked piece of RNA, which infects plants. And what's less than RNA? Proteins like prion proteins, right? Which are called spongiform encephalopathies. Then we moved on to talk about animal viruses. And remember, all viruses follow the same 5 general steps in order to replicate and multiply. But there are 2 important differences when it comes to the animal viruses versus bacterial viruses. And the important differences occur at penetration and release. Remember that the bacteriophage drills a hole through the cell wall and injects the DNA. But an animal virus, the entire virus is engulfed and moved into the host cell, usually by receptor-mediated endocytosis. But the entire virus goes inside. The capsid will break apart, right, to release the nucleic acid, and so the infection can continue. When it comes to release, here we have another important difference. We just said that bacteriophages use lysozyme to completely rupture the bacterial cell wall. Certain animal viruses will also rupture the host cell wall, but others steal host membrane, right? They bud from the host cell, exocytosis. And we said that potentially more viruses can be produced this way because the longer the cell lives, the more new virus particles that it produces. But eventually the host cell will die because it is losing membrane, and host cells can only synthesize and replace that membrane at just so fast a rate. But the main difference occurs at penetration and release. We know that some animal viruses can activate oncogenes, which turn normal cells into cancer cells or tumor cells. And one of the things that happens is that those cells lose contact inhibition. And so they start growing on top of each other, forming those tumors. If the tumors become malignant, they will metastasize and spread. So there are certain families of viruses that contain examples of oncogenic viruses, but remember the entire family is not oncogenic, just certain members within that family. Some animal virus infections can be latent. Latent viral infections are sort of analogous to lysogenic, bacteriophage infections, except that latent viral infections, you are infected with the virus, it periodically goes dormant and then reemerges, and then goes dormant and reemerges. And we talked about herpes viruses being the best example of this, where you may have genital herpes or oral herpes, so you get blisters, they heal, right? They disappear. You may go for months, weeks, months, or years without getting another outbreak. But the herpes viruses are always there, right? They're hiding in nerve cells, and they just wait, and periodically they reemerge. A persistent viral infection is one where the immune system cannot control the virus at all, so it persists. The virus continues to replicate, it continues to do damage, ultimately ending in death most of the time. The example we talked about was subacute sclerosing panencephalitis, which is caused by a measles virus. But a persistent infection, the virus is still there, it's still multiplying, it's still doing damage. It just progresses over a period of time.
Are those types of viruses common or not so much?
Oh no, not very common, right? Fortunately.
Yeah, that's good.
Now there are 8 known human herpes viruses, and you need to be familiar with all of them. Herpes 1 and 2 causing the oral and genital herpes, herpes 3 causing a chicken pox, 4 the Epstein-Barr virus, 5 cytomegalovirus, the 2 roseola viruses, and the number 8 the Kaposi sarcoma virus. What they all have in common, they're all herpes viruses. They're very common. Most people are infected with multiple herpes viruses. They generally are very easily spread through saliva. So make sure you review those. So I already talked about latent infections with herpes. When it comes to the families and genus and species, so in the handout, the things that are underlined are the most important. So make sure you go back and study those. How do we control or treat viruses? Well, you can rely on your immune system. If your immune system is able to successfully fight the virus, then you survive, and you're immune to that particular strain of that virus. But we do know that viruses mutate and change. We can use vaccines made of pieces and parts of viruses or maybe dead viruses. And what they do is they cause the production of antibodies. And so you guys, if you've been listening to the news, you may have heard of Pfizer's vaccine, which they claim has showed to be at least 90% effective. And it's actually a nucleic acid vaccine, and specifically an RNA vaccine. And so the idea is they inject you with the RNA, some of your cells will absorb it, and they'll translate it to make those viral proteins, and that's how you create the antibodies against them. So you're making the proteins, and your immune system makes antibodies against them. One of the things that I'm curious about, they've been playing with nucleic acid vaccines for about 30 years. not one of them has ever been approved for use in humans. So this would be the first nucleic acid vaccine approved for use in humans. So I'm just curious to know why exactly no other nucleic acid vaccines ever been approved, and any concerns about this one. So we have to wait and see, but hopefully it works as well as they say. But transporting the vaccine, storing it, and delivering it is more complicated since you need extreme cold, like minus 70 degrees Celsius, to store it. And it has a very short shelf life, so distribution is going to be the main problem, but hopefully it will work as well as they say, and then we can start getting back to normal sometime next year, probably by the middle of next year. But those are vaccines. We also have certain antiviral drugs, but as I mentioned, especially in the case of influenza, if we start overusing these antiviral drugs, they're going to stop working because the viruses will mutate to become resistant. But we do have a variety of antiviral drugs for different kinds of viral infections. So for coronavirus, remember I talked about 3 related viruses, SARS-1, SARS-2, and MERS. Right, so SARS-1, we had that kind of little outbreak back in the early to mid-2000s, and have not seen that one since. We're currently experiencing the SARS-2 pandemic, right? We're living through that right now. MERS is primarily confined to the Arabian Peninsula, in particular Saudi Arabia. Most of these viruses appear to have a common original host, most likely bats, and then ultimately they get transmitted to humans through other animals. So SARS-1 and SARS-2 were first documented in China specifically. As we know, the viruses were transmitted to humans from some other animal. Most commonly, coronaviruses are transmitted by droplet infection, coughing, sneezing, even just breathing can release microdroplets. And it's the primary mode of transmission. Secondarily, by touching contaminated surfaces and objects. What do we call those contaminated objects which can spread infection? What do we call inanimate objects that may be contaminated?
Fomites.
Right, fomites, right? Although that's not really considered a major source of transmission, but it's definitely possible. So the symptoms, make sure you just review the symptoms. Most of the symptoms, and we're focusing on SARS-2, are similar to, colds and flu, shortness of breath, difficulty breathing, loss of taste or smell. That does strongly suggest that it's not the flu and maybe more serious like COVID.
Professor, I have a question, please.
Yes? Mm-hmm.
The COVID-19, which of the tests is more effective? Is it the antibodies or the PCR?
So which of the tests are what now?
Is more effective?
Well, they both can give you false negatives. The problem with the antibody tests, they have a lot of difficulty with those to start with, and antibodies may not be produced that strongly, at least not early in the infection. and the other one actually looks for the viral nucleic acid, right? So they're actually looking for the actual virus in your nose. And so they both, the one that's easier is the nasal swab, right, to do that test to look for the actual virus. The antibodies, you have to give a little bit of blood for that, to test. And again, both of them have false positives and false negatives, although usually it's false negatives. I don't know whether the antibody test is as good as it should be yet. I haven't checked up on that recently, but the nasal swab tests are getting very good. So, but the nasal swab test just tells you if you have the virus right now. And the antibody test can tell you if you had it in the past, but may no longer have it. So they both give you different kinds of information.
Can you be positive for antibodies and have the virus at the same time?
You can. There are some people who, for some reason, even though they've recovered, they're still shedding the virus. And so they still have it in their nose and sinuses. So they're still infectious, even though they're perfectly fine, right? They have no symptoms because they actually recovered and they're fine, but they're still shedding the virus. And so they're still infectious. And then, of course, you have the asymptomatic cases, which have the virus and may not have produced any antibodies yet. So, there's no guarantee if you go get a test, and it's most likely going to be the nasal swab, that all that's going to tell you is that you're not infected right then. So,
Right.
but Maryland actually does want people to go get tested. If you go search Montgomery County website, you'll find a schedule. It's free. You may have to schedule an appointment, but you can still just kind of walk in and get it done. But they actually do want people just to go get tested so they can get a better understanding of how many cases there really are in Maryland. So if you have a chance to do that, go check that out so you can go get tested for free. But it is the nasal swabs. It'll just tell you if you have the virus right then and there, at that particular point in time.
I heard they might close down the state again. Is that true?
Say again?
I heard they might go back to phase 1 on Friday or something. Is that true?
I haven't heard that. I know some states are talking about doing that kind of thing, having a shutdown again, because, cases are skyrocketing right now. With all the, Trump rallies, and then you had, protesters and people celebrating, and this, people are just sort of letting their guard down now. And so the case numbers are actually skyrocketing in the United States. And the number of deaths is also going up, not as fast as the number of cases, because we're able to treat patients better so they survive, better now than they did back in February and March. So,
But Professor, how long can the virus stay in the nasal cavity or in the nose?
how long can it stay where?
How long can the virus stay in the body?
Again, it depends on the person. As I mentioned before, there are some people who got infected and recovered, but they're still shedding virus. And so they're still infectious, even though they have no symptoms and feel just fine. So it depends, right? They just don't know. Most people, if they get infected and they recover, they should be fine, not infectious. But other people, even when they recover, are still infectious for weeks afterwards, right? So they just don't know. It just depends. And that's why, like the professional athletes, if they test positive, they're quarantined for 2 weeks, and then they have to get 4 consecutive negative test results in a row after that, right, to know that they're no longer infectious. So, because some people stay infectious longer than others, even though they fully recovered. Right, so there's no simple answers here. So symptoms typically take 2 days to 2 weeks, right, to show up, but that's sort of an average. Of course, we all know how to prevent transmission, right? Masks, washing hands, social distancing. And of course, there are many controversies, which you can go back and review. When it comes to disease, remember we talked about concepts of disease. Again, there's terminology for you to know, like pathology, right? The study of disease, the etiology, the cause of diseases, pathogenesis, the development of disease, infection, the attempted colonization of the body by pathogens, and a disease, which is an abnormal state of health caused by that pathogen. And we know we're all heavily colonized with microorganisms, right? They become a part of us permanently. They're called the normal microbiota, or I still like the old term normal flora. And they do live in symbiotic relationships. They may be commensal organisms where they benefit, but they don't help or hurt us, or mutual organisms where we both benefit. Koch's postulates, remember there are 4 of them, and these are a series of steps to perform to identify the cause of a particular disease. We know there are problems with the first 3 postulates. The first postulate, the suspect microbe is always found in diseased individuals, never healthy ones. Of course, we know that's not entirely accurate, right? Because carriers may be perfectly healthy, but they're carrying a pathogen that they can spread to others. But assuming, that the suspect microbe is only found in diseased individuals, you then try and grow it in the lab. Again, this is not always possible because we don't know the growth requirements for every pathogen. But assuming you can grow that pathogen in the lab, you use your test culture to infect a test animal. And then that animal should manifest or reproduce the same symptoms of the disease. Again, this is not always possible. Many pathogens are very host specific, so you might not be able to find a suitable, test animal to use. But assuming you can, that suspect microbe should be actively reproducing inside that test animal. And if you follow these 4 postulates, you should be able to conclusively identify the cause of a particular disease. Diseases that can be transmitted are communicable. If they're easily transmitted, they're contagious. And if they cannot be transmitted, they are non-communicable diseases. We do find some diseases in populations always, but in very low numbers. So we call those diseases endemic. If that disease spreads to a larger percentage of the population, it becomes an epidemic. If it spreads all over the world, it's a pandemic. And then we talked a little bit about herd immunity, where if the majority of the members of a population are immune, that protects those members who are not immune. Because if the majority of the population are immune to a particular disease, it's very difficult for that disease to spread through that population. And that protects those members who are not immune. A localized infection, we typically refer to bacterial infections this way. So it just occupies a very small part of your body. If it spreads to a much larger area, or spreads throughout your body, it becomes a systemic infection, which is potentially very serious. Often you have a primary infection like a respiratory infection, cold or flu, which leads to a secondary bacterial infection. If you find bacteria in the blood, we call that bacteremia. Not unusual or uncommon, but your immune system should quickly eliminate any bacteria that enter the bloodstream. If your immune system can't, and the bacteria are actively reproducing in the bloodstream and producing toxins, then you have septicemia, which is very, very dangerous. If you find those toxins in the blood, that's toxemia, viruses in the blood, viremia. And subclinical disease, that refers to a carrier who is asymptomatic. So a person who has no symptoms of a disease but is actually carrying a pathogen and can spread that pathogen to other people and make them sick. And what was the classic example we talked about of a person who transmitted disease even though she had no symptoms?
Is it the COVID-19?
A classic example from a long time ago.
Was it that Irish Mary?
Yep,
Her name?
right. Typhoid Mary.
Typhoid
Right.
Mary.
Typhoid Mary, right? She was infected with Salmonella typhi but had no symptoms, but she was able to transmit that disease to other people, right? Some of whom died. Diseases can be transmitted through contact transmission, right? Touching, kissing, sexual intercourse, and that's direct contact. Indirect contact requires an inanimate object, which we said was a fomite. Coughing, sneezing are droplet infections to spread over usually no more than about a meter or so. If you want to spread the infection over a much greater distance, you need a vehicle like soil or water, like food or water. Vectors can transmit, so insects, right? They can transmit diseases over greater distances. We talked about mosquitoes and flies, and even people, right, can be a vector to transmit diseases. What is a nosocomial infection?
Hospital infection?
Right, so hospital acquired, and primarily from the people that are in the hospital, right? So what kinds of things can be done to reduce nosocomial infections? Well, wearing protective equipment, making sure doctors and nurses wash their hands or change their gloves in between patients, disinfecting things like stethoscopes, wiping down and disinfecting surfaces. So it's all about, good hygiene and keeping things clean. Incidence, remember, is the number of new cases relative to the total population size at a given point in time. Prevalence is both new and old cases in that population. And so incidence will tell us about your relative risk of contracting that disease, whereas prevalence is more of a measure of the burden of that disease on society. It can also tell you how widespread that disease is. And so remember, incidence and prevalence, whether they're high or low, does depend on the duration of that particular disease. So if it's a short duration disease like colds and flu, you may have a very high incidence, but a low prevalence over time, right? Because people recover from colds and flus quickly. But we talked about something like lead poisoning, where you do not recover quickly. So even though there may be no new cases and the incidence may be low, the prevalence may still be high because of all those old cases that are still there. And so to calculate, remember, incidence, number of new cases divided by the population size, and prevalence, new plus old cases divided by the population size. And when it comes to pathogenicity and non-specific defenses, remember, only pathogenicity will be on the exam. Non-specific defenses will happen on exam 4. So only 1 through number 7 of these objectives. All the rest will be for exam 4. So again, we have terminology, pathogenicity, right? Virulence, infectivity, and toxicity. So make sure you review those. Remember, there are, 3 portals of entry. Technically, it's more like 2, right? One is mucous membranes and the other is skin. But if you inject directly through the skin, that's sort of the third portal of entry. But mucous membranes and skin. And of course, the portal of exit is related to the portal of entry. So, for example, a respiratory pathogen, you inhale it through droplets, and then you cough and sneeze to release more droplets. Or skin-to-skin contact for things like herpes or staph infections. The fecal oral route, you eat it, and it comes out the other end. So the portals of entry and exit are related to each other. Once the pathogen gets inside of you, it will produce a variety of virulence factors, which help it to survive, spread, and cause damage to the host. So adherence factors are sticky things like the glycocalyx and fimbriae. A variety of enzymes might be produced. IgA proteases, collagenase, coagulase, so different enzymes. And these all help a pathogen to survive and spread. Christiana, can you turn your microphone off?
My microphone is off. It's
Oh,
not
yours
me.
is off? Oh, whose microphone's on? You need to turn it off.
Sorry, that was
I'm not
me.
the only one with babies.
All right. Yeah, I'm not looking at the Blackboard screen, so I can't see whose microphone was on. So toxins, we know that many toxins are produced. If a pathogen has the ability to make a toxin, it's toxigenic. If we find toxins in the blood, it's toxemia. We can use those toxins to our advantage by neutralizing them and using them as a toxoid vaccine. And then antitoxins are preparations of antibodies, which we can use to neutralize toxins. Exotoxins are actively secreted by bacteria. Endotoxins are only found in gram-negative bacteria, the outer lipid membrane, in particular the lipid A portion. But remember, it's not very toxic, right? We talked about LD50. If something has a very high LD50, that means it's not very toxic. But if it's got a low LD50, then it is very toxic. Enterotoxin is a specific type of exotoxin that acts on the gastrointestinal tract to cause diarrhea. And remember, the pathogen is doing this on purpose because it's trying to spread, right? It wants to get out to find other hosts to infect. And then super antigens are not super good, they're super bad, right? They cause an overreaction by the immune system. Excess amounts of cytokines are released, which do damage to the host. And so super antigens can cause a lot of damage because the immune system actually does the damage to the host. And so that's the end of the material for exam 3. So any other questions?
Professor, is there going to be any bonus question?
Yeah, there'll be a bonus.
Except that they are all the answers, answer keys, right?
And what do you mean?
I don't know what to call it. Objectives.
Yeah, no answer keys for the objectives. But I covered almost everything, right?
What I mean is it going to be the same format as
Yep.
it's used to be? Okay.
Multiple choice and a couple of short answer essay type questions. Anything else?
So do you think the vaccine will work? The one that they're coming out with?
Well, I hope it does. I mean, the early results were promising. So if it works as well as they say it works, then it'll be good. And there's another vaccine, a very similar vaccine by another company. So if one works, then the other one should work too. And so the more companies, the more vaccines we have, the more options we have. So like I said, maybe by next summer we'll be close to being back to normal.